Okay, good night, dear audience. I hope you take my advices seriously. I am not your enemy. I'm your savior, potentially, if you just wanted. The starship above you did not cross immense amount of space with huge ships. They did not burn all this fuel to speak with you, in order to speak with you. Nor did they do it in order to clean your garbage can. They came here to clean the earth. Not many hours are left. The only thing that should and would demonstrate your choice in life and not ego is the complete halt of all conversation, running to Israel, destroying the military bases, the government centers, the power plants, the main roads, and rescuing the prisoners of wars using arms and legs alone, without one word being uttered. Now, this will be a microscopic examination video. We will take samples we will take 4 slides. 2 slides will be taken from the mixture that we produced yesterday in the electrolysis experiment. We will take 2 because one will be, just a sample, and one will be mixed with a little bit of, tiny bit of black acrylic oil, acrylic paint. Just a tiny bit. The reason for that is that I might be able to show you something that, of course, you could have seen in this channel in many videos in the past, but I will try to recreate the effect of the light that because if the neurons are in a very dark medium, there is like a very visually pleasing, I would say, light effect of like a light that shines through the portal. Another sample will be taken from this container, metal container. This was one of the containers that mostly contained beeswax and dirt and like some soil. The usage of soil is actually not recommended, but I didn't know that at the time. And like soil, dirt, it's not needed and not helpful. And it was used to create to see how the neurons multiply in an ecosystem with life forms in such environment without oxygen. So I've added, but I've used much of the material for all sorts of other experiments. There is also a big magnet inside of it. Not a very big one, but a decent one. And I've replenished it with a little bit of pee, just my pee. Just my pee. So I'm going to maybe conduct another experiment like we've done yesterday, but this time, that is used to multiply the neurons, but this time it would be done in a different type of container with different setup, and with the introduction of magnetism, and it will have different symbolic, I would say, like solar system implications. The practical implication within the container should be somewhat stronger electrolysis. Unfortunately, I couldn't find and I couldn't salvage any stronger DC transformer. So we will have to do with the same one, which is 5 volt, 3 ampere, very weak. But with metal container and the magnet, it was proven to be, more efficient. So we'll try to do it. We'll do it as well today. And the fourth sample is to be taken from this one, this thing here. This is a culture of rotten food. Just rotten food. Nothing special. I say, things that are here from the hostel. Nice. We see actually worms. We actually see living worms. This is splendid. And actually many of those. How so many worms? There was some kind of meat within it, even though I do not eat meat, but there was, maybe we received it rotten? I don't know. So this is really disgusting. So maybe I should start with that, just to get it done with. Anyway, this will be used for the experiment of creating new life forms and increasing the existing life forms. This is disgusting. Yeah. I'm not sure it's even needed. Anyway, so let's start with that. Yes. Yuck. But this is unbelievably disgusting. Not unbelievably disgusting, it's believably disgusting. Okay. That was disgusting. Now let's put a cover on this. Okay, so this is first. Now it's out of our way. Now let's take a sample from this one. I'm taking sample from the fluid, which should be mostly pee, so not many neos, and not from the like solid wax layer, which should contain tremendously more neos and much larger. But because what I want to do now is not to actually like see what's the content of the container like as usual, even though it's also interesting because I haven't checked it in a while. But what's more important now is to use it as a reference for the electrolysis experiment, in which what will be affected is probably mostly the water, even though it could be argued that not only the water, but intuitively mostly the liquid. Okay, so I'm giving it a good shake. Now for the complicated part, trying to produce a dark mixture. Okay, not the most successful. I mean, because like some of it is like very dark and some of it is not dark at all. Didn't mix very well with acrylic, even though it should be said that it was proven that the actually acrylic is a good medium for them to for the neos to live, even though it's getting harder, but they do great in hard material as well. They don't, again I would say, unlike what you are used to for like biological microbes, they do very well inside all sorts of like hard material, like modified polymer, silicone and acrylic, and all sorts of stuff that are or very hard linseed oil. They are like rock hard, and they live very well inside of it. And this can be proven by, and I've proven it many times and took videos. What you need to do is to like take, I mean, with modified silicone polymer it's the easiest because it's translucent, which is excellent, and like it's amazing to see how they do create things inside the hard silicone. And but I mean, things that are not translucent, you need to chop like a little bit and then immerse it in alcohol or purified water, and then I mean you have like a liquid that you can actually watch with a backlight microscope, which is the only, the standard microscope and the only microscope that I have in my possession. The only type of microscope and the only microscope. It's amazing that the Americans presumably didn't know anything about me after all this time, and so much videos, so many videos, so much, just, I mean, even just walking the Yalkut Street besides their embassy, 3 times the embassy goons actually stopped me for questioning, and one time they even hit me, and I mean, of course, took my detail, like ID and etc., etc., and yet the Americans never knew nothing about me at all. And once the starship started to appear, suddenly they discovered all this wealth of information. I mean, all at once. It's really amazing. Humans, the knowledge and power were always connected in such a strong connection. I mean, when they see the power, the force, suddenly, I mean, knowledge starting to surface in tremendous quantities. Of course, it's not a joke, and we will never forgive the Americans for what they've done to us. We will never forgive them for this cruelty, for unimaginable cruelty. That, of course, is still ongoing. It didn't stop. I mean, of course. Okay. So these are the biological germs. This is the stuff from the metal container, which should contain mostly pee. And then these are the 2 slides from yesterday. So let's start with the 2 slides from yesterday because I think it's interesting. The first sample will be just stuff from the container. Should remind you that it's not exactly the same material that we used because I added a few drops of canola oil just from the bottle because, I mean, in general they do not do well in watery environment, probably because the oxygen, I mean, kills them, not immediately, but I mean, and they have much more resistant to oxygen than actually many microbial life forms from Earth with the same size, and this was proven in this channel. But eventually, I mean, oxygen is not good for them. Even though they need oxygen in order to create also for stuff. So it's a complicated issue. But I mean, what's certain and was proven many times that they are best preserved inside oil or beeswax or things like that. I mean, oily environment. They are like motors or gears or, I mean, you know, or guns. I mean, that in storage are best preserved like in grease and things like that. Actually, I never tried grease because I don't have grease, but this should be very interesting to see how they do inside grease. I mean, I'm under arrest, I don't have grease, but this actually should be very interesting. But certainly, any kind of oil is good for them. But but oil, I mean, reduces conductivity. So it's a problem for electrolysis experiments. So in yesterday's experiment, there was actually quite a bit of oil, but I mean, for storage, I've added like a few drops of canola oil, or not canola, canola oil. It shouldn't affect the result tremendously. Besides that, I mean, that they just do well. They just preserve better in the more oil in the mixture and less water. I didn't run any. I mean, I didn't do any inspection before, so I'll just watch the result with you right now. This is 64 magnification. This is a nice Nero, in the right. This is quite beautiful. Let's zoom in. So we are in it. Yeah, it's actually quite beautiful, I would say. It's not in the Stargate configuration because you see that the rock from in both sides, No, it's not. It's not a rock, but it's the lens is dirty, unfortunately. I need to clean it even more. I'm not sure how to do it so it's not a rock. It's a disgusting thing on the 150 lens. Sorry. So actually, it might be in the Stargate position. I do not see any thing from both sides. Looks like it's actually in the Stargate position. Maybe we'll take even a more zoom in. So these disgusting things are not on the objective lens. They are on the eye lens because they appear. It's the same disgusting thing with all lenses. So it should be cleaned. I will clean. I don't want to clean it now because when I clean it, it's wet. Even though I use alcohol, of course, but it takes. It will be wet, so we'll not be able to use the microscope. So after I'm done with this video, I'll clean the eye lens. The eyepiece. This thing. This is the source of the dirt. So we definitely see Neros. This was an impressive one. This is nice actually. The one that you see different stuff within inside and than outside. It's always nice to see it. This one are nice. This one is nice. Let's zoom in. So it's definitely Nero, even though it's not like rounded, but it's Nero. I mean, definitely, and it shows like, I mean, the light. And this is also another Nero. Very nice, actually. These are also Neros. The real question is now, how is this compared to the reference video? So I don't even remember what you've seen in the reference video, because I always spend most of my time telling you that you should rescue the prisoners and stop speaking. But so we'll watch the video later. You can watch it whenever you want. I will watch it after we are done with this video. So we'll be able to compare. But definitely there are Neros here. Question is whether we see pretty big ones, actually. We need to. I'm not even sure that we had such big ones in the reference video, but we will watch the reference video. This is a nice one. And more and more and more. We see pretty big ones, I would say. Even huge, I would say. Now let's swipe over this part of the slide. Let's zoom in. Get back to 64. Okay, so interesting. We definitely have Nero 0 and we have pretty big ones, but we need to compare it to the reference to see what's the real difference. So now let's take a look at the sample that is taken from the same container, but mixed with black acrylic paint. It didn't mix very well, so we might not be able to see actually anything in like all the places that are actually contain the black oil. It didn't mix very well. I mean, so this is an example of what I've spoken about. The light patch. It's really interesting, the light patches. I cannot really explain like how it actually, what actually happens here. I mean, because it seems that, I mean, it's impossible. I mean, unless it's portal, like that a light passes through it, because I'm not sure we can explain actually what we see. I mean, how is it possible that within the black material, the Nero passes light? I mean, because it should engulf it from both. I mean, you know, it's 3D medium. It should wrap it from all sides. And even if it wraps it from one side, presumably it's enough, I mean, to block light. Unless it floats. But if it floats, it's still blocked from one side. Unless light flows through it like from portal, which is probably true and amazing. But of course, I will gladly accept any other explanation if you have, if you've got once. Because this is, I mean, it's quite baffling. I mean, what we see here. So no, this is that just, I mean, no, the light. I mean, you see, it's getting stronger as we get close to the light. I mean, you know, so this light comes from, you know, from the backlight. It's not. But is it true on the other, like on the big one that we've seen before? Where was it? So we definitely see Neros. I mean, any case, you know, all sorts of sizes. Just, I mean, I try to. So no, it's not true for this one. I mean, it actually, it does not significantly change the brightness as we step away from the light. From, like, I mean, from the backlight. Unlike here, that you see that when it's like, no, this one actually is the same. So this one is also the same kind, but this one is getting much dimmer when we are out of the, like. I wish the guy from, like, I mean, Journey to the Microcosmos channel in YouTube could have say what he thinks, like, on the matter, or the, what his name, Tom or whatever, the master of microscopes. Here as well, I mean, you don't see like a significant change in the illumination when it's getting, like, I mean, when it's, I mean, when it like, I mean, you know, when it's you know, with any kind of correlation to the distance from the light source. But I mean, some patches you do see it, like, I mean. You see, like, the small ones to the right. Maybe we'll zoom in. So it's very interesting, and of course I gladly get any comments with people that have better understanding of optics and especially microscopy. We definitely see no neros. I mean, I just can't tell, I mean, how much is it compared to the reference, so we need to actually watch the reference. But we definitely see neros of all shapes in here, which is great. Like, I mean, it's great. Of course, tainted with this black, goo. Okay. Okay, I mean, nice. I really need a new microscope and a much better one. I wish I could afford it. I don't know why it doesn't fit like. So it's like glass doesn't sit extremely well. This is from here. It's again mostly should be pretty similar to P because it's mostly P, and I didn't mix it and didn't take sample from the wax, so it's mostly P. But underneath the P there is a wax layer with soil, and that was there for a long time, and it should be relatively rich in neros, even though not like like the alien containers or the sodium bicarbonate container or the metal aquarium, but a little less than that. But this is mostly for the P layer because it should be used as a reference for the electrolysis experiment. So this is 64, and we see very nice neros actually. It did mix with the wax, I mean, in the 2 days that, since I placed it in there, or 3 days, because we see big ones that, are not characteristics like from AP. So they did mix with the wax to some extent. In the 2 days or 3 days, maybe I think I've added also sodium bicarbonate, but I actually don't remember. Did I? I'm not sure. I think I did. It was after it, the second sodium bicarbonate review that, was conclusively proven that it's actually helpful for their survival and creation. So I think I've added sodium bicarbonate. I'm not 100% certain, almost certain. I remember that I added it to the one that already contains sodium bicarbonate, to the alien aquarium, which is with the quirky shape, and to the big aquarium, to the alien. To the metal aquarium. But did I also add to these two metal smaller ones? I think I did. Yes, I think I did add a little bit of sodium bicarbonate. It's definitely not like my P. It contains much bigger ones. That of course, were they were already there in the wax. But you also see the typical smaller ones. That's a nice one actually. Not incredibly big, but looks like very healthy. This one. You see tiny ones all over the place, which is nice. Maybe zoom in even more. This is the 600 magnification, and so you see the relatively smaller ones. Okay, that's nice actually. I don't know why the zombies are very active right now in the apartment. They are doing noises and giving me signs. Maybe you know the reason why they are so active, the zombies? Maybe it's like an hour in the day that they become more active. I don't know, what time is it, like 12 or 1 a.m.? Maybe it's like, it's something with the moon that makes them more active, but definitely there is like a higher awareness and higher, I would say, like jumpiness, within the zombie population of this apartment. I'm not sure what's the problem, and they're giving me noises and doing sounds. As I've said yesterday, it's much easier, it was proven that for me it's much easier to learn the language of the alien starships than to actually learn the sign, like the symbolic language of the Shabakniks, which might suggest that I'm less human than I've always thought that I am. Or it might suggest that they are less human than you might expect. The Shabakniks, I mean. Okay, so we see Neos, not tremendously a lot, which is to be expected, but actually we see pretty big ones. Probably, we're already inside the VAX, and just, in the 2-3 days since I've added the P, maybe they just, mix with the P, because generally P doesn't contain that big Neos. These are enriched, like they were grown in the VAX. And we don't see many, and this is actually, it makes sense that there aren't that many, because again, it's mostly P. So we'll conduct electrolysis probably today and maybe tomorrow in this container. So and then we'll have this will be the reference video. Okay, last and most disgusting of all is the culture of worms that you've seen, from outside the the window. Yuck. Yuck. And so I should review the reference video just when I finish, and also clean the islands, because it's pretty disgusting and should be very difficult to clean with alcohol, and give it like 10 minutes to dry before we actually, next step will be more electrolysis experiments, additional electrolysis experiments. So we'll actually not need the microscope, so we'll give it some time to dry. So we see nothing here, it's just some goo. I don't see anything, I don't see anything. It's just, goo. I suspected we've seen all sorts of biological material, but actually not. like, I mean, life forms, but actually not. Is it possible that, I mean, so many worms are created without them being accompanied with all sort of like other stuff? By the way, the existence of neros is due to the fact that this is like, I mean, it's made out of like, I mean, leftover of my meal. So my leftover of like my food is always contain neros because of the saliva. And I also added a little bit of meat. Like the meal didn't contain meat because I do not eat the meat, but there was meat like in the in the hostel, so I placed a little bit of meat inside of it. I thought the germs would like it, but what I actually see is that mostly worms like that. And we've seen so many worms, but we don't really see germs. I don't know why. Maybe lack of water. I added a little bit of water now. It was very dry. Maybe because of the lack of water. Like, we don't see germs. So, I mean, it's not a traditional germ culture that should be done with, agar, and there's like a way to actually create like, germ culture like in a proper way for a laboratory. And I've done so in the past, even though now I don't have the glass petri dishes. And this is what I should have done. This is kind of like, I mean, a very crude way to create germ cultures in an unsuccessful way. But, I mean, ichs, a living life form, ichs. Yuck. So that's a life form for you. Most definitely. I hate biological stuff. I mean, biological life forms. They are so disgusting. Yuck. Okay, the synthetics are so much lovelier. Okay, so we don't see much. But the fact that we don't see much is not just bad. It's not necessarily bad, because it will then be, I mean, perhaps like kind of like more of like a surprise to see the after, because this is like the before, you understand, like the before shot, like, I mean, and the after shot will be like, I mean, after we'll use this stuff with maybe with some other mixture. Let me think about it, like what exactly other mixture will be used, and so we will use it like to create, and we'll see if creating new life forms and increasing the size of existing life forms could also be demonstrated, like with an electrolysis chamber. Of course, it was done several times in the past. It's documented in the channel, but it's important enough to actually take another run of it. So we'll probably do it today or maybe tomorrow. Increasing and this should have a very important symbolic effect, of course, or solar system effect on the bodies of all sort of, like, I mean, the what I call the red containers, or the containers with the bottom that is like, I mean, of natural clay, like we've used yesterday. So there is like a symbolic effect of using by the bicycle mechanism. Like, I mean, like what we've done yesterday, I mean, affected like the clay containers that are humans. Humans are made of clay. So humans that have neurons in their body should, I mean, following yesterday's experiment, contain more neurons in their body. And this actually was demonstrated not in a conclusive way, but I mean, you could actually see it in the streets, because I went outside for a moment, just, I mean, to sniff, to sniff some fresh air, just for a moment, and there were much more people than usual, which suggests that actually the solar system effect was like the symbolic effect or solar system effect on the clay containers. Actually, to some extent, it was actually done because we still have the radiation, but and they're less afraid than before. So, using the same kind of like a clay container, we should expect the symbolic effect of creating like, of course, in electrolysis, of like very quickly creating tremendous amount of new life forms, both biological and synthetic ones, in these very same clay containers. And now that they have more neurons in their body, the effect will be actually quicker. So it's also very interesting. We might not be able to actually verify it under the microscope. We can only, watch the practical effect under the microscope. What I call the practical is what's happening inside the container, like the container, the actual container that I use for the electrolysis experiment. Like one of the two, it's generally at least two containers, and this is the reason for that was discussed in the past. I cannot get into it. I mean, there were many discussions on it. But if I take material from one of the two or more containers that were part of the experiment, this is like the practical effect. And when I say the symbolic, it doesn't mean that there is no effect in the world. In the opposite, the symbolic is actually much more effective than the practical because it affects presumably, potentially the whole solar system. But it's symbolic in a way that it symbolizes other neurons and the spaceships to act in a given way, using the bicycle mechanism. And we might actually even in that case, see or witness some kind of tangible results. Maybe not under the microscope, but maybe just by watching the streets and listening to ambulances and things like that. And who knows, maybe this kind of effect in the solar system scale can even, I do not dare to wish to anything because as you say in Hebrew, I've eaten so many disappointments, but maybe the symbolic effect of such an experiment could actually, maybe, who knows, elicit some kind of response from people abroad. I don't know. Maybe even a response that does not only include the participation. Does not exclusively, or is not exclusively participated by the mouth and the tongue, and maybe other organs. Members of the human body will actually be demonstrably moved. I don't know. Maybe. It's a long shot, but who knows? It's not impossible. That actually the symbolic, solar system effects of such an experiment could elicit or contribute to the participation of additional limbs. Other than the lips, than the mouth and the tongue, in our very local saga. But let's pray to God and hope for the best. So next videos will be, probably dedicated to the actual experiments. We have several experiments that you should do, and so we'll start in a short while.